investigations into the microbial food web. Previously, we've learned that the microbial food web at its present moment includes both the microbial loop, viral loop, together with the classical food chain. But how did all these components were investigated in order to come to the conclusions that we have now? So let's look at the first main components, the primary producers, the phytoplankton, in relation to the main grazers of phytoplankton, which are the microzooplankton. So a very simple and elegant experimentation to investigate both the potential production by the phytoplankton and the grazing by the microzooplankton is via the Landry acid dilution method. Here the sample is filtered by a 0.2 micron filter to obtain particle-free seawater. The acronym is PFS. And a series of bottle incubation microcosms is set up with 0.2 dilution, 0.4, 0.6, 0.8 to 1.0 dilution fractions. So for 1.0, that means the whole microcosm is filled with the sample, whereas for 0.2, the ratio is 0.2 of sample with 0.8 of particle-free seawater. Because the grazing by microzooplankton upon phytoplankton is primarily through interaction, so the principle of the method is, as the sample gets diluted, the distance between the predator and prey increases. So the chance for the microzooplankton to actually graze upon the phytoplankton decreases as the samples get diluted. Therefore, experimentation will show that the production in all these bottles increased with reducing sample. So the production here is the ln difference in chlorophyll at time 0 versus time usually 24 hours after incubation in 12-hour light, 12-hour dark situations. So theoretically you should get a slope that's going up like this. Of course, this is impossible to obtain. So usually we set the highest dilution at 0.2, and from here we can extrapolate to intercept at y-axis, and this indicates the potential production, because this indicates the production of phytoplankton when there is totally no grazing, whereas the slope is equivalent to the grazing rate. So from this Landry acid dilution experiments, we are able to obtain both potential production by the phytoplankton and also the grazing of microzooplankton upon phytoplankton. So this is a very elegant experiment that provides both this set of data. So from here we realize that both phytoplankton and microzooplankton are actually coupled together when phytoplankton production increases, microzooplankton grazing also increases. So other than these two components, the main heterotrophs are actually bacteria. But however, we realized from studies that bacteria in the sea has different mode. The main modes of living is actually either free living or attached mode. So usually the criteria used is either less than 2 micron or less than 1 micron filtration, which means for samples that pass through the filter, the bacteria is denoted as free living, whereas those bacteria that are trapped on the filter, they are attached. So usually for an experimentation like this, samples are collected and then count for total bacteria using a direct count stained with DAPI to give you total bacteria concentration in the sample. Then the same sample is also filtered, either less than 2 micron or less than 1 micron, and then the filtrate is used to carry out the DAPI direct count, and the bacterial count in this fraction is denoted as free living bacteria. So from these two components, we are able to calculate for attached bacteria abundance in your sample. So where the total bacteria minus the free living bacteria will provide the attached bacterial abundance. Here we can see a line graph showing one of the methods to depict the data available. So the y-axis is the bacterial abundance, and let's just say hypothetically the x-axis shows the distance offshore. If the total bacteria count here shows higher counts near shore as opposed to offshore, which is quite logical because near shore has more anthropogenic impact, has more nutrients as opposed to offshore. Whereas for free living bacteria, we can obtain, and if this line shows the free living bacteria, so the difference between this is actually the attached bacteria. So this graph can show the difference, all the three components: total bacteria, free living bacteria, and attached bacteria. Although the relationship between phytoplankton and bacteria is not direct, but it's actually through an intermediate DOC pool, where we know that up to 50% of primary production can be exudated out as a soluble organic matter into the DOC pool. And this organic carbon is mostly utilized by bacteria. Only very few organisms other than bacteria can utilize DOC directly. So the main component is usually bacteria that utilizes the DOC and is incorporated into its biomass, which is then taken up by protozoa and then back to microzooplankton of the classical food chain. So this is the microbial loop that we've talked about. So because of this indirect relationship with where DOC is the intermediate, meta-analysis have shown that this coupling is actually very strong and is exhibited in various habitats around the world. So if the bacterial biomass on the y-axis is plotted against the chlorophyll A concentration, you will see a straight line correlation that shows the strength of the coupling between bacteria and chlorophyll A. After the consumption of DOC, bacteria undergoes bacterial production to produce more biomass, and this bacterial biomass is actually top-down controlled by protozoa. How can we measure this process? So one simple way is by using size fractionation, where we incubate the total seawater in one microcosm, while the others will incubate at less than 2 micron. So the filtration is to remove the grazers. So what we have here is a portion of the sample where we have bacteria and the grazers, and another portion where we have bacteria alone without grazers. So at less than 2 micron. So when we plot the change in bacterial abundance on the y-axis over time, we will obtain a positive slope, which can be denoted as mu of growth rate. What we will observe is that the growth rate, the bacterial growth rate in the less than 2 micron fraction should be higher than the total fraction, because here the bacteria has grazing pressure, is under grazing pressure, is under top-down control, whereas here the bacteria grows without any top-down control. Therefore, from the difference between these two slopes gives you the grazing rate, where the mu for the less than 2 micron fraction, you minus the mu in the total, that will give you the grazing rate. So now we know how to evaluate the relationships between phytoplankton and zooplankton, and also between bacteria and protozoa, and the different modes of bacteria, whether as free living or attached bacteria in the environment. So now let's look at the viral loop. Here we know that bacteria, other than protozoa, bacteriophages also controls bacterial population. However, the virus loop is not so clear-cut, because viruses can adopt two different modes, either a lytic mode or a lysogenic mode. So if it's a lytic mode, the virus will infect the bacteria, then lyse the bacteria, and when the bacteria lyse, there is contribution to the DOC pool. However, in lysogenic mode, the bacteria will infect the virus will infect the bacteria and then integrate into the bacterial chromosome and becoming a lysogen. And only on certain occasions, environmental factors that will induce lysogens to revert back to lytic mode. So in experimentations where we want to know whether viruses are adopting lytic or lysogenic mode, we use the compound mitomycin C, which induces lysogeny into lytic modes. So what we can do is we can carry out two microcosm experiments one as a control and one where mitomycin C is added. So here we know that we can count the virus-like particles by direct counting on anodis. Anodis is about 0.02 micron pore size, and stain with the cyber gold fluorochrome stain. So if you were looking at just the virus-like particles over time, you will see virus-like particles increasing because of viral production. And because mitomycin C induces all lysogen into lytic mode, naturally the amount of virus-like particles should be higher in this microcosm. So in an experiment where we try to see the differences in virus production at different temperatures, you may obtain a graph like this, where virus production increases with temperature increase, which is very logical because as temperature increases, biological processes also increase. However, in a mitomycin C incorporated incubation, you may see a different pattern, also increasing but maybe at a different rate. Okay, so at low temperatures you can see that the gap between mitomycin C induced and control is large. However, the gap decreases with temperature. So this shows that as temperature increases, there is less lysogens, because the difference between mitomycin C induced and the control narrows. So less lysogen in at high temperatures. So this is how scientists can find natural factors that induce lysogens to adopt the lytic mode. For example, higher temperatures and higher growth rates. So this is